Named Lucky, my lab cat. And let me make sure this is up and running. Everybody always waves. I love that. So Lucky and I spent a good month or so together because we would do a little bit of a system. Like we got very involved, way more involved than y'all. Y'all gonna do your pig in a day. Like this cat was weeks and weeks. So then you had to pack the cat back up, put it back in the fridge, and then come back, and then you just kept revisiting the cat. Right. So my cat was so young and so pliable that a lot of like the tissue dissection, like going in between muscles or whatever, you could do with a finger, like a gloved finger, you could just run and it would just break the connections and you could see everything. Whereas my friend with the older cat, like she was like digging stuff apart. But here's the crazy thing, her cat was a total mutant. And it was wild because no, like it had my professor calling other college biology professors all over the state of Virginia and being like, can I email you pictures of this cat? Because her cat had an esophagus and it went slightly off center, like where it's supposed to go through the diaphragm. It was slightly off from where it was supposed to be. And then it had like no stomach and a small intestine like small intestine like thing. And then it had like a little bit of large intestine. So it was lacking most of its digestive tract. And one of its lungs was like shriveled up and the other one was too big, and then its heart was offset and its heart was very small. So my biology professor, Dr. Goddard, was like, this was not a cat that lived outdoors its entire life. This was a pet that got out, because she's like, there's no way this cat could hunt. Its heart was too small, its lung capacity was off. She's like, this cat could not have hunted and kept itself alive. And we knew it was old because its teeth, you could tell by its teeth that it was an old cat. So that was fascinating. She had the first most fascinating cat. And then my little Lucky was the second most fascinating cat, because when I opened up Lucky's stomach, it was full of worms.
Oh yeah.
They're preserved too, like the cat's preserved, so the worms are preserved. But they were like kind of white or off-white, and they were all curled up in there together like a big knot. Like knot.
Hey, can we wait till after we eat, please?
No, that's part of the biology. Sure. So
It's okay.
So, like,
this last time.
I was like, excuse me, Dr. Goddard, but I got worms. And she's like, let me see. So she goes, oh, take them to Harriet. So Dr. Gray, Harriet Gray, was my advisor. And she was this, at that time, little old lady. Now she's even more. But she was, we all thought she was a little old lady. She was probably in her early 70s at the time, but she was early gray and, gray hair. And she wore leather loafers, stockings, a wool skirt, a turtleneck or a button-down blouse, and a cardigan and glasses and hearing aids. She looked like your grandma. And she was fabulous because she liked to make us uncomfortable at every opportunity she could get, because she did not look like the type. So, but her focuses were like neurobiology and entomology, so she knew her insects very well. So, and her worms and whatever else. So I had this gloved hands full of worms. Now, Dr. Gray was about 80% deaf. So to be a woman in the 90s with a PhD in neurobiology and be 80% deaf, she overcame some things. Like she for real did in her younger years. So the problem was, in her office, she sat facing away from the door, facing the window. So you could stand behind her and dance a jig, and she would not know you were there. Like she would turn around and write on the whiteboard, and we'd all start talking. And then she'd look at us and we'd be like, because she can hear. So I walk in and I've got like worms. And I walk up behind her and I'm like, you know? So she'd spin around, get a tap her, and she looked at me, and I just held them out, and she said, from your cat. Because she knew what we were doing. And I'm like, yeah. And she's like, stomach or heart? So I told her stomach, and then she immediately spat out whatever the genus and species of worms were, and I toted them back into the comparative anatomy lab and announced to the class what I had. And everybody was like, Dr. Gray's like the coolest person. But Dr. Gray would do wonderful things like, I'm just going to give you one example of this woman, and you're going to indulge me here because you're all fairly grown people. But in invertebrate anatomy, we were talking about snails. And this woman liked to make people, she loved to make us all uncomfortable. Women's college, right? No dudes around. We're all just sitting there. And she's talking about snails and snail reproduction, and she's writing on the board, and her voice would get more quiet. And everybody would lean in, and this chick spun around with a dry erase marker in her hand and said, and some snails have wonderfully long penises. which the entire class went. But she did that on the regular, and it's one of the things I loved best about her is that she intentionally would like catch us, and we'd all be like, "What is happening?" But she because she was a neurobiologist, she got all the experimental hearing aids too, because she could report back to the audiologist with information at a level that other people couldn't, right? So they gave her these brand new cutting edge hearing aids to try, like a beta research thing, like she got to wear them. And she announced to everybody, these are new hearing aids. That's what she talked very quietly. These are new hearing aids. And the special feature is that if it gets too loud, they will just cut off. And we're all like, okay. So she turned around and write on the board, and we would all talk. It's what we'd always done. And one day somebody said something, and we all started laughing, and she spun around. What happened? What happened? And we're all like, She's like, God, dead quiet. What happened? So we were laughing. You also had to use TDD to call her. Anybody ever use TDD? So TDD is the service where you dial on a landline and you get a translator who types to the deaf person on the other end because she could not hear through a phone. So if you ever had to call her at home, and she started every class by warning everyone, call me anytime you have a question. If you're studying for a test, whatever, you call me, you have a question, you will have to talk to an interpreter who's going to type for me and I will type back. But beware, the interpreter will type everything he or she hears, everything. So if your friend's in the background going, this is easy, I got the answer from so-and-so already, or ask her to postpone the test, I'm gonna hear it. So we all knew, like, she was amazing. I absolutely adore her. All right, so now we're getting ready to roll. So we're doing cell processes today, right? All right, so cellular respiration. This is what happened in lab today, a type of anaerobic respiration, and you've got aerobic respiration in your cells. So energy is released by breaking what? What has to be broken? Caitlin.
Chemical bonds.
Chemical bonds. We know that breaking bonds releases energy. Forming bonds requires energy, right? So what is a measure of heat energy? And it's defined as enough to raise the temperature of 1 gram of water by 1 degree Celsius. What's that? A calorie. Very good. All right. Now, calories are interesting. We're going to talk about calories here in a second. So what can, let's just say store, sorry, more energy than carbohydrates or proteins? Fat, right. All right. So your fat's going to store more energy than carbohydrates or proteins. So chemical energy and food. ATP synthesis occurs 24 hours a day, every day of your life. Now, this is something interesting to consider. When you look at, like, what's your Capri Sun say for calories? 90 calories. So calories with a lowercase c is the measure of heat, which is enough energy to raise the temperature of 1 gram of water by 1 degree Celsius. Calories with a capital C is kilocalories. So how many calories is it? 1,000 calories. So your 90 is 90,000 calories, actually, with a little c. It's calories with a big C. That's a kilocalorie.
1,000.
Where?
1,100. It's 100.
Oh, you're right. I'm missing a zero. You're correct. Oh, you caught that. Write that down. So the calories you consume will only indirectly provide your body with the energy it needs. They actually provide the energy to produce the high energy bonds of the ATP molecules that then power your muscles and energy for your brain's electrochemical responses. When you take in fewer calories than you need in a given day to run those systems, the body turns to fat stores, to a lesser degree, protein from existing muscle, to convert carbon compounds into ATP through cellular respiration. Drew, you were talking about this the other day. What's this law?
Oh, the first law of thermodynamics.
It's the first law of thermodynamics. You eat fewer calories, you're not going to have as much, but you can't burn it, right? So you've got to basically, this is one of those cut calories to lose weight situations. I saw a video the other day, and it was actually titled "What I Eat Every Day as a Fat Girl." I don't make that up. She wrote that. But she was a larger lady, and she was talking about how she really doesn't eat all that much. She's like, I really don't eat all that much. Well, she maybe didn't eat all that much, but what she ate was very calorie dense. She's like, here's my smoothie and here's my whatever. And I'm like, ooh, so whatever you're taking in, if you take in more calories than you can burn, your body's going to convert them right into fat. But if you take in less calories than you need, your body's going to go get it from fat. If your body runs out of fat to get it from, then it's going to take it from your muscle. Unless, interestingly, babies are born with a different kind of fat. They have something called brown fat that exists only in infancy. It grows out of us, but it is there as an evolutionary adaptation. So if something happens to mom and baby's not getting food, there's nobody to feed the baby will go into its brown fat stores, which burn kind of at a different rate. So it is a way to ensure that the baby can keep living. So it's interesting. We don't have brown fat, not anymore. We're all past our brown fat stages. So we have multiple steps in respiration. We have glycolysis, pyruvate oxidation, which they call the transition cycle. We have the Krebs cycle, which is also called the citric acid cycle. And we have oxidative phosphorylation, which is chemoosmosis or electron chain transport. So a lot of different names and a lot of cycles that interact with one another. All right, so go. This is what happens in each of them. Glycolysis is anaerobic, so does it need oxygen? No, right. And basically what's going on here is you're breaking down two of your sugars into two pyruvates, and you're going to release some ATP. And then in this next part, your pyruvate is going to get converted to acetyl-CoA, and you're going to release carbon dioxide. So it says right here, it's oxidative decarboxylation. So you know there's oxygen involved. And we're releasing carbon dioxide. What do we exhale? Carbon dioxide. Okay, this is where it's coming from, partly. Then you have your Krebs cycle. This happens inside your mitochondria. And this is when you oxidize acetyl-CoA to CO2, which is what's kind of happening over here. And then you release ATP and some other guys.
I have a question.
Yeah.
So in the diagram it says Krebs, but in the is Krebs
Citric acid.
Is Krebs named after a person?
Yes, and I'm gonna show you who he is in a minute. Yeah,
If not, then it would be apostrophe S.
Yeah, it's named after a person. You're right. Very good. That's observant. Good. And then you have this electron transport. So this is in your inner mitochondrial membrane, and it's going to use your NADH, which you produced up here, and your FADH2, and you're going to create this electrochemical potential with an H+. So now you've got an electrical gradient. So many things in life, in the world, all comes down to electricity. We learned this last year in chemistry, right? Like atoms interact with other atoms based on their charges. Like it all comes down to some bit of electrochemical involvement. And we'll go over that more in a minute. I'll show you. So glycolysis, if we take glycolysis and look at it more closely, in glycolysis, which is a 6-carbon sugar, what is it? Somebody say glucose is our 6-carbon sugar, and it's going to undergo a series of chemical transformations. So here's our glucose right here. You see it? So in the end, it gets converted into 2 molecules of what? And, a 3-carbon molecule. What do you think? Pyruvate, yeah, right here, right? Which is a 3-carbon organic molecule. And in these reactions, what is made? And NADH plus is converted to NADH. So what else is made? Yes, ATP. How many are made? 4. All right. Glyco means what do you think?
I guess.
Yeah.
Sugar.
Yeah, good guess. All right, so glyco means glucose or sugar or a sugar derivative. And what's lysis again? We've been through this one before. What's lysis mean? Yes. Breaking. Okay, very good. So to split or break apart, glycolysis, glycolysis, breaking apart a sugar. And in the first step, our 6-carbon sugar is broken apart. So that's why it's called glycolysis. So how do we oxidize this oxidation of pyruvate? Okay, so what's going on here? So each pyruvate goes from glycolysis, goes into where? Where does this happen? Physically, where does it happen? So it goes into the mitochondrial matrix. Remember, we always say that mitochondria are the powerhouse of the cell, right? Well, this is why, because this is where the energy production is going on. The innermost compartment of, anybody know where? In a mitochondria, right? And it's converted into a 2-carbon molecule bound to a coenzyme A, known as what? Do we know? Do you know?
Is it acetyl CoA?
Acetyl-CoA. Very good. Acetyl-CoA. All right. Carbon dioxide is released, and then what is generated here? Can we tell? Carbon dioxide is released, and here's your NADH, which is generated here. All right, these are all steps of your aerobic respiration. All right, here's your Krebs cycle. So there's no apostrophe here because sometimes you'll see it with it, and it is appropriate, and sometimes you'll see it without, which just because it's become a thing. So acetyl-CoA made in the last step combines with 4-carbon molecule and goes through the cycle of reactions, ultimately regenerating the 4-carbon starting molecule. ATP, NADH, and what are going to be produced? FADH2. And what is released? Can you tell what's released? Caitlin?
ATP.
Carbon dioxide, which are good. That's all right. It's multiple cycles, right? Okay. So you asked about Krebs. All right, now this is one of our fun, interesting moments in science history and world history. No. This is Sir Hans Krebs, the biochemist. Okay.
Is he German?
Yeah.
But this is
Oh,
General Hans Krebs, not a biochemist. The biochemist, he's a Nazi. Okay, but here's the interesting thing. This guy was Jewish and he fled to England because of this guy with the same name. Like people like him, right? So we have 2 Hans Krebs in history. If you look up Hans Krebs, you will find them both. You need Sir Hans Krebs, not General Hans Krebs. General Hans Krebs and his ilk ran Sir Hans Krebs out of Germany into England, and you can tell he is Sir, which means he has been what? Knighted. So he ended up in England. That's why he's Sir Hans Krebs. He's not going to be Sir Hans Krebs if he's in Germany. That's not going to be a thing. So yeah, so they wouldn't let him practice medicine or do anything else, so he got the heck out. So that's one of those interesting historical moments. If you look up Hans Krebs, you are going to find them both. So be sure you know which one you're looking at. You want the dude with the Harry Potter glasses. You don't want that. It's not you. That's not a good guy. Yeah, that's not, am I wrong? He has Harry Potter glasses, right? I mean, come on. They're cute. They're cute little glasses. So we have oxidative phosphor. Well, the whole Harry Potter narrative, the whole Death Eater narrative is a World War II Holocaust narrative. I mean, it's all analogous. If you've ever, I don't know if you've ever really thought that or paid attention to it, but the whole Death Eater thing is absolutely analogous with Nazis, like intentionally so. Yeah. So the whole mudbloods, think about it, right? Okay, so chemiosmosis and electron chain. NADH and FADH2 made in other steps deposit their electrons where? And the where? In this electron transport chain, which is exactly what it sounds like. We're going to move the electrons down the chain, right? So turning back into their empty forms, NAD plus and FAD, you notice they've lost what? What did this lose? NADH lost a what? Which is a hydrogen, and it's a plus now, right? So it has a charge. FAD lost its H2. So look here, our H pluses are what we're dealing with as far as electrons transporting, right? We have that a lot, right? Especially in chemistry, we talk about it a lot, how you get high H plus, right? You get this little hydrogen ion. I asked the juniors today, what was the only element that didn't have a neutron? And I was bummed that nobody answered. Please, somebody tell me. Hydrogen does not have a neutron, right? So if it's an H plus, then what's it missing? Hydrogen has one electron and 1 proton. If it's H plus, what's it missing, Drew? It's missing the electron. So essentially, what is it? No, yeah, it's just proton, right? Yeah. Isotope has extra what? It's an ion because isotopes have a different number of neutrons. Very good. Very good. All right, good. Okay, so now let's see. As electrons move down this chain, energy is released and used to pump protons out of the matrix. When you see words like that in biology, pump, you know you're dealing with an active, not a passive process. So just keep that in mind. You'll see that a lot. So, and then that forms a gradient. So here's your gradient. You have all of these positive ions floating around and you've got a gradient. Now your electrons are going to be attracted to them, right? So protons flow back into the matrix through an enzyme called what? Here they are flowing back. What's this called? There you go, ATP synthase. All right. And that makes ATP. At the end of the electron transport chain, oxygen accepts the electrons and takes up protons. And then what do you get? Oxygen, water. Think about it. Think about it. You were so excited though. But think about it. If you've got these little guys floating along here, and they need to pick up oxygen, accepts them, and then takes up protons. You got negative, H negative, I mean, well, electrons, and you've got these little hydrogens in there, and they've got water. They're going to come together. I mean, sorry, oxygen, you're going to get water. All right, so here's your overview. Glycolysis is 10 steps in the cytoplasm. Pyruvate oxidation is 1 step in the mitochondria. Krebs cycle is 8 steps in the mitochondria. And electron transport chain is a multi-step process in the mitochondrial membrane. So this takes place in and around the mitochondria, right? So let's see if we can kind of figure this out. Which one requires oxygen, anaerobic or aerobic? Everybody together. Aerobic is going to require oxygen. What do you think evolved first, aerobic or anaerobic?
Anaerobic.
Who says aerobic? Hands. Who says anaerobic? All right. And then mitochondria are going to be involved in which process? Anaerobic or aerobic? 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 There you go. Fermentation. We did this in lab today. We talked specifically about why it's fermentation. Anaerobic. Very good. Electron transport chain is going to be where? Aerobic. And then the Krebs cycle. Aerobic. It's right here. Right here. Okay. Yep. All right. So here it is summarized by our favorite amoeba sisters who are beyond adorable, right? They will summarize it nicely for you. Oh, here's a diet coach.
Sponsors make such great.
Okay.
That is not on the list.
Dang. That's
Really?
crazy. Why are they so fun?
Are you a morning person? One of us is, and one of us is definitely not. Mainly because when I wake up in the morning, it just takes a while for me to get my energy back. It takes a lot of time and coffee for that to happen for me. Cells really don't have that luxury. They are busy performing cell processes all the time. Active transport of many substances needed for their survival, for example. And the energy currency they need, specifically, is ATP. ATP stands for adenosine triphosphate. It's a type of nucleic acid, actually, and it is action-packed with 3 phosphates. We have a video all about ATP and how it works as an energy currency. So where am I going with this? Well, cells have to make this ATP. It doesn't really matter what kind of cell you are. Prokaryote or eukaryote, you have to make ATP. The process for making that ATP can be different, however, depending on that type of cell. One way is called aerobic cellular respiration. Lots of organisms can do aerobic cellular respiration, but this video is specifically going to go into aerobic cellular respiration in eukaryotic cells. That means this video is talking about the process within cells that have membrane-bound organelles, such as a nucleus and mitochondria. Eukaryotic cells include the cells you find in protists, fungi, animals, and plants. The mitochondria, which can be found in most eukaryotic cells, are going to be kind of a big deal in this aerobic cellular respiration because some of the process occurs in the mitochondria. So let's get started. Remember, the major goal for any organism performing this is to make ATP. Okay, here's the overall look at the equation for aerobic cellular respiration. Remember that reactants, inputs, are on the left side of the arrow, and products, outputs, are on the right side of the arrow. This equation, by the way, looks remarkably similar to photosynthesis. Look at how the reactants and products are on different sides. Well, that doesn't mean they're simply opposites. It does show that they have substances in common. In photosynthesis, organisms make glucose. Notice how glucose is a product. But in cellular respiration, organisms break the glucose down to make ATP. Fun fact, you know how when a bean seed first germinates in the ground, it isn't able to do photosynthesis yet? Yeah, the germinating bean is relying on glucose that it has stored, and it's doing cellular respiration to break it down to make ATP so it can grow. Of course, once it starts to mature and develop leaves, it can do photosynthesis. It will be able to do both photosynthesis and cellular respiration. Plants make glucose in photosynthesis and they break it down in cellular respiration. They can do both. But if you aren't photosynthetic, such as a human or an amoeba, you have to find a food source to get your glucose. You need glucose to get this process started, and we're going to assume that we're starting with one glucose molecule. Step 1, glycolysis. This step takes place in the cytoplasm, and this step does not require oxygen. It's considered anaerobic. In glycolysis, glucose, the sugar from the equation, is converted into a more usable form called pyruvate. Glycolysis usually takes a little ATP itself to start out. The net yield from this step is approximately 2 pyruvate and 2 ATP molecules, and 2 NADH. What is NADH? NADH is a coenzyme, and it has the ability to transfer electrons, which will be very useful in making even more ATP later on. We'll get to that in a minute. Now, an intermediate step occurs. The 2 pyruvate are transported by active transport into the mitochondria, specifically the mitochondrial matrix. In the mitochondria, pyruvate is oxidized. The 2 pyruvate are converted to 2-acetyl-CoA, which will be used by the next step. Carbon dioxide is released and 2 NADH are produced. Step 2, the Krebs cycle, also called the citric acid cycle, still in the mitochondrial matrix. The citric acid cycle is considered an aerobic process. While the cycle doesn't directly consume oxygen, some of the events in the cycle need oxygen to continue. To learn more about this intricate cycle where the 2-acetyl-CoA will enter, check out the further reading suggestions in the video details. Carbon dioxide is released. We also produce 2 ATP, 6 NADH, and 2 FADH. FADH is also a coenzyme, like NADH, and it will also assist in transferring electrons to make even more ATP. Step 3, the electron transport chain in chemiosmosis. This is just a beautiful thing, really. In eukaryotic cells, this is still in the mitochondria, and to be more specific, it's involving the inner mitochondrial membrane. We do require oxygen for this aerobic step. This is a very complex process, and we are greatly simplifying it by saying that electrons are transferred from the NADH and FADH to protein complexes in electron carriers. The electrons are used to generate a proton gradient, as protons are pumped across to the intermembrane space. All these protons being pumped out into this intermembrane space generates an electrical and chemical gradient. The thing is, if you remember from our cell transport video, ions, like the H+, don't easily travel across membranes directly without something to travel through. The protons can travel through an amazing enzyme called ATP synthase. If I could be any enzyme, I'd be ATP synthase because it has the ability to make ATP by adding a phosphate to ADP. ADP is a precursor to ATP. ADP has 2 phosphates, but if it obtains a 3rd phosphate, it becomes ATP. So, in chemiosmosis, the protons travel down their electrochemical gradient through a portion of the ATP synthase, powering it to make ATP, the ultimate goal. Oxygen is the final acceptor of the electrons. When oxygen combines with 2 hydrogens, you get H2O, water. If you remember from our equation, water is a listed product. Now, the electron transport chain in chemiosmosis step makes a lot more ATP compared to the other 2 previous steps. How much? So I've noticed in my years of teaching and the assortment of textbooks I've collected over the years, there are some different numbers in cellular respiration charts. In fact, you can even see them change a bit in different editions of the same.
Yeah, it's
And
real
it's
fun.
made me want to emphasize that it's important to not just memorize them, because it really is more of a range. I want to focus less on the number of ATP made per glucose molecule because it depends on a lot of variables. One variable is the gradient we were talking about. How many protons were pumped across the mitochondrial membrane? You can see more variables discussed in the factual references. And those references have estimates ranging from 26 to 34 molecules of ATP per glucose molecule in the electron transport chain and chemiosmosis step alone. Then, if you add the other two steps, Krebs, otherwise known as the citric acid cycle, and glycolysis, you could estimate a range of anywhere between 30 to 38 net ATP molecules total per molecule of glucose. Again, to emphasize a range. Now, this is just one way of creating ATP. But like we had said at the beginning, all cells have to make ATP, but the way they do it can differ. If there's no oxygen available, some cells have the ability to perform a process known as fermentation. It's not as efficient, but it can still make ATP when there isn't oxygen. We have a video about that. We can't emphasize enough how important the process of making ATP is for cells. For example, cyanide, which is found in some rat poisons, can block a step in the electron transport chain, which would block ATP production. A poison that prevents ATP from being produced can be deadly. With the important role that mitochondria have in ATP production, there is also a demand for increased research on mitochondrial diseases. We are confident that the understanding of how to treat these diseases will continue to improve as more people like you ask questions. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So it's complicated. I saw a funny meme the other day on Instagram, and it was some doctor talking about how he was glad to get out of med school so he didn't have to keep memorizing and reciting the Krebs cycle. It's that complicated that even people that like go into that field get tired of it. It's like, ugh. Okay, remember our friend, Miss Winnie Sloan?
Sure the pool.
Yeah.
It says
Where?
out, There's out.
Out. Oh, write it down. Write it down. Write it down. There you go. She's awesome. And she has one here for this process. And as always, really good. This is the
Okay,
quick one.
all respiration starts out with the same first step, glycolysis. It's universal, it's anaerobic, it begins the oxidation of glucose, which means you're taking the energy that's stored in the bonds of glucose and transferring it into something the cells can use, hopefully ATP. Now, along the way to do glycolysis, you actually have to spin 2 ATP to get your party started, but you get 4 back, so you net out 2 ATP. And it also reduces to NAD when you do glycolysis. Now, your step after glycolysis depends on if there's oxygen present or not. So glycolysis, though it's shown as one step right here, it's really 10 steps and 10 different enzymes. But one of your end products is going to be pyruvate and to reduce NAD and to ATP. Well, the ATP will get used up, that's great, but you have a finite amount of NAD, and if all of it is in a reduced state due to glycolysis, you're gonna run out. So you have to have a way to oxidize it. And fermentation allows you to do that anaerobically. What you'll end up doing to oxidize your NAD, you have to reduce something else, so you'll reduce your pyruvate into either ethanol, if you're a plant, or into lactic acid if you're an animal, and that gets your NAD oxidized. But if you do have oxygen, there's a better way because there's more energy left in that pyruvate that you can harvest from. So aerobically, we'll go a different pathway. So following glycolysis, aerobic respiration, you will continue to oxidize your pyruvic acid, but now into what's called acetyl-CoA, again forming more reduced NAD. Those are starting to accrue, and that's good. So then you'll take your 2-acetyl-CoA and put them into the Krebs citric acid cycle, which will complete the oxidation of your glucose. And you're going to generate, as a result of 2 turns of that, 6 reduced NAD and 2 reduced FAD and a couple of ATP. This is great because now you can take all of your reduced NADs and FADs to the top of your electron transport chain located on the cristae, the inner membrane folds of the mighty mitochondria. There, you can drop off your high energy electrons, thus oxidizing your NAD and FAD so they can be reused again. And now these high energy electrons are going to get past this electron transport chain, which along the way, some of the coenzymes can help you concentrate hydrogen ions and take them from the matrix and push them out into the inner membrane space. And there you will concentrate hydrogens, which is a difference in concentration, charge, and pH, all of those hydrogen ions are gonna wanna come back into the matrix, and the only way they can get in is through ATP synthase complex, thus generating a whole bunch of ATP. But the only way you can keep that chain moving is if at the bottom of the chain you have oxygen to accept those final electrons and keep everything going.
So of course, she's got another song. So, so I'm not gonna have you go through and write out all the steps like we did last time. If you find her stuff helpful, go back and watch her videos after class. But here she goes, puts the whole thing together.
Cellular respiration. Okay, so what is the first step called? Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm. What do you start out with? Glucose. How many carbons? 6. What are we gonna do?
Break it in half.
How many steps?
10.
How many enzymes? 10. What are these called? 2 pyruvate acids can do reduce some NAD and make some ATP. 2. Oh, we have plenty of oxygen. What should we do? Aerobic respiration. What's the next step called? Transition. Right. Here we go. And you reduce some NAD and 2 acetyl-CoA, put 1 away. What's the next step called? Krebs cycle. What's another name for it? Citric acid cycle. How many times are you gonna do it? 5. Why? 2 acetyl-CoA. And when we say the words do the bit, what are we doing? Pulling off the carbon dioxide and we're reducing a NAD. Here we go. 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP. Reduce an FAD. Reduce an NAD. And 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP. Reduce an FAD. Reduce an NAD and stop. Gather all the reduced NADs and FADs and take them to the top of the ETC.
Electron transport chain.
Reductase, cytochrome oxidase, and you make some ATP. A lot of it, a lot of it, a lot of it, a lot of it, a lot of it. Who catches electrons at the bottom?
Oxygen.
Forming
Water.
Okay, so now we're gonna do aerobic respiration. The first part is still the same. We're still gonna do, right? And we end with nucleic acid. And so glycolysis is the thing, but then we're gonna say, oh no, we don't have any oxygen. What are we gonna do? Fermentation. How many choices do we have? 2. You and I make lactic acid. Makes our muscles sore. Plants and yeast make
Alcohol.
Either way, you oxidize your NAD.
She is incredible. Like, that's wild. So there are other ways of making ATP from glucose without oxygen, such as anaerobic respiration and fermentation, which we just talked about. 2 kinds of fermentation are lactic acid fermentation, which takes place in your body, and alcoholic fermentation, which plants and fungus, right? So after you exercise, sometimes anybody ever get almost like the crunchy muscle feeling? Not just sore, but it's sore, yes, but it's also crunchy almost. That's lactic acid. That's from lactic acid fermentation. You didn't have enough oxygen for the work you were doing. Huh? Okay. So cellular respiration versus photosynthesis. The energy in photosynthesis and cellular respiration flow in opposite directions. Their chemical equations are opposites, which she talked about in the Amoeba Sisters showed you, but there it is, right? 6 water, 6 carbon dioxide give you a glucose, and 6 oxygen molecules, right? Same thing at the bottom, except it's the other way around. And it gets very complicated. There's another example of how it all looks together. All bound together, all the things working together. So, anaerobic bacteria do not require oxygen for growth. By the way, I'm wearing bacteria today.
Yay.
Aren't they cute? Yes. So anaerobic bacteria do not require, excuse me, oxygen for growth. Anaerobic cellular respiration is similar to aerobic cellular respiration in that electrons extracted from a fuel molecule are passed through the electron transport chain, driving, what are we synthesizing? ATP. Some organisms use, what is this? Oh, do you know? Nobody knows what this is? Nobody from chemistry wants to give it a guess? Do you want to give it a guess?
Something sodium, right?
Is that sodium? That's not sodium.
Sulfur.
It's sulfur. Sulfate. That's sulfate. SO4 2 negative. We did sulfate a lot last year. As the final electron acceptor at the end of the transport chain instead of water, you know. While others use nitrate. Yeah, I was just going to give you that one there. Sulfur or another variety of the molecules. Okay. So fermentation or types of fermentation. Our first type is alcoholic fermentation. This takes place in what kinds of organisms?
Bacteria.
Plants, yeast, things like that. So you generate your NAD plus so that the glycolysis can continue to produce ATP. However, alcoholic fermentation in yeast produces ethyl alcohol instead of lactic acid as a waste product. Alcoholic fermentation also releases carbon dioxide. It happens with the yeast in bread, beer, wine. Plants can do it too. And then you have lactic acid fermentation. And this is when your NADH transfers its electrons directly to pyruvate, generates lactate as a byproduct. Lactate, which is just the depronated form of lactic acid, gives the process its name. This happens with certain bacteria like Lactobacillus acidophilus, which is what creates yogurt. It happens in muscle cells that also carry out lactic acid fermentation, though only when they have too little oxygen for aerobic respiration to continue. So when you've been exercising very hard all day, even if you're panting, you're not getting enough oxygen to those muscles, they will undergo lactic acid fermentation, which you can then feel later. Your muscles get sore.
So you work out and you feel pain.
Yeah, well, that's one of the reasons. It's one of the reasons why your muscles can be sore. Anybody know the other? Why your muscles are sore after you work out? Why? You know.
Oh, like bearing?
Yeah, yeah. Like if you're
You know,
lifting weights or whatever or doing
you don't eat enough protein, it won't be able to recover itself.
right. So that's why you eat protein after a workout because you actually are tearing your muscle fibers. That's how like people that work out a lot get big muscles because you keep tearing those fibers and then they rebuild. So they keep filling in more muscle cells everywhere they've torn, they get bigger and bigger, right? So you get glucose, pyruvate, and your different ways you can break down for your fermentation here. You have aspergillus that can make stuff like lactic acid for soy sauce. You get lactobacillus that can give you stuff like cheese and yogurt. And then these guys over here, you can end up with beer, wine, bread, all that fun stuff. So if you look at the processes side by side, you'll see they're very similar up to this point where you have aerobic pathways where there's oxygen versus anaerobic pathways. And otherwise they start the same way, right? Help her out. There you go. So science of beer. Didn't you always want to know about the science of beer? Okay, well now you do.
What website do you want
Yeah.
with Wix?
Yeah, I love Wix.
For your work?
Please. There you go.
Nothing like enjoying a beer on a hot weekend
You don't.
afternoon or
None of you
get the
do.
taste of the alcoholic drink. There's actually a ton of science that goes into creating it. Science may be the last thing on your mind when enjoying a cold one, but it's always good to know just how what you're enjoying was made. Science contributes to the type of beer, the different flavors, and how it's alcoholic. So let's learn the science behind brewing beer. First, we'll look at the ingredients. The main ingredients in any beer are water, malted grains, hops, and yeast. Hops are the flowers of the hop plant, which are used for flavoring. The only other uses for hops are in herbal medicine. With these four ingredients, you have everything you need to make beer, but the combinations and processes make all the difference. All of these ingredients are boiled together for set amounts of time until the final mixture is filtered and sent to fermentation. Mineral contents in your water source can make a huge difference to the type of beer that you brew. Calcium and magnesium in your water, otherwise referred to as hardness, affect how the yeast grows and metabolizes the sugar in your brew. Bicarbonate, or HCO3, in your water also affects the pH of the beer during the fermentation process. If you're wanting to brew a beer that is more caramely or toasty, then you'll need to roast your grains more than usual. Darker roasted grains result in grains that have more degraded glucose molecules, ultimately resulting in a darker beer. Hops in beer are the main driver behind modern beer, and without them, our pints would be a little less enjoyable. The hops act as a surfactant that allows bubbles to be trapped.
Surfactant, I don't know what you
Beer. Hops aren't added to the water, it would have much less surface tension, resulting in no head and a bitter taste. Hops can provide certain flavor notes to the beer, but their main effect is stabilizing the brew to allow our palate to taste all of the different bitter and sweet notes of the beer. Without hops, we would have a hard time tasting the flavors and ultimately enjoying the beer. Next, we move on to fermentation. Yeast is the big player in this process, and it's the most important element of making it alcoholic and carbonated. These little cells seek out glucose molecules in beer left by the grains and hops. By fermentation, the yeast converts the glucose into ethanol, or alcohol, and CO2, which is the carbonation. The specific chemical equation is what's shown right here. This is the general equation for fermentation, but specific products can be influenced by the type of yeast used in brewing. Ale yeasts are top fermenting, which means they can ferment at higher temperatures and ultimately produce more esters, which are organic compounds easily described as fats and oils. Lager yeasts are bottom fermenting, which means that they ferment at lower temperatures, producing a crispier taste. Wild yeasts are seldom used in the brewing process, but when they are, they produce a mixture of compounds that are more acidic and an acquired taste. These are rarely used because the exact flavor profile is harder to control from a brewing perspective. During the fermentation process, certain molecules can have different effects on the final flavor and feel of a beer. The length of carbon chains in alcohols, the amount of alcohol, the type of esters, residual sulfurs, and carbonation levels all contribute to the final flavor profile of the beer. Factors that can influence the fermentation and alcohol content of a beer are mineral content, temperature, pitch rate of the yeast, aeration, and length of the fermentation process. Brewers closely control all of these variables to get flavors that they want out of the final brew. Esters produced in the fermentation process can be predicted and controlled to produce certain flavors as well. Ethyl acetate is the most common ester in beer and smells like nail polish remover. In order to smell this ester, a high content needs to be present in the beer, and this usually isn't the case, which is why your beer doesn't smell like nail polish. Ethyl acetate is another ester that smells of bananas.
That
This
one
ester
does
is
smell
found in
exactly
many
like banana.
Belgian and wheat
That was
beers.
not.
Ethyl butyrate gives a hint of passion fruit or pineapple to the beer, bringing up the sweeter notes. Finally, another ester that you probably have tasted or smelled is ethyl hexanoate, which results in an apple-like flavor added to the brew. As for the science behind popular beers, we can dig into some of the most popular beers to see just how precise every aspect of the brewing process is controlled and how it's made. Budweiser is a Pilsner malt, meaning a lighter golden beer with a sweet hint and medium amounts of hops. They use a type of yeast named S. overum and ferment their brew at 15 degrees Celsius for 2 weeks and add wood chips during the fermentation process to help the yeast grow. Finally, the beer is heavily filtered and forced carbonated. Guinness, a darker beer, has dark roasted malted grains that give the color and light roasted malts that provide sugar for fermentation. They use a yeast named S. cerevisiae and ferment at 18 degrees Celsius for 2 or 3 weeks. The beer is then filtered and carbonated with nitrogen, which results in the creamy taste due to nitrogen creating smaller carbonation bubbles. Finally, another beer to look at is Rodenbach. It has lighter roasted malts and wheat with low to medium hops. They use wild yeast with spontaneous mixed cultures to ferment at 18 degrees Celsius for 2 to 3 weeks. The beer is then filtered and fermentation continues in the bottle to add natural carbonation. Smaller breweries will use natural carbonation in the brewing process because the end product can vary slightly and filtering is needed less. For larger breweries like Budweiser, filtering is a large aspect of their production, which removes any natural carbonation, so they must add in carbonation after the fact. For many, the taste of forced carbonation versus natural carbonation makes a huge difference in the beers that they enjoy. After all of this science is completed, you get a final brew. There's really no reason to store beer for long periods of time like other alcohols because the flavor profile is already completed at the time of brewing. In fact, most beers do have an expiration date of within a year, even shorter if natural carbonation is used. So be sure to drink up, but of course, responsibly.
Or not. But there's there's so much science that goes into that. A ton, like everything from the type of wheat you use to whether you force carbonation later or not. So like we make cider here, like we've got it on tap in there. And ours is a combination of natural and forced carbonation. So you get some natural from the yeast releasing carbon dioxide, but then we put it in the keg under pressure, which will then again increase more, add to the bubbles, basically. So it's all science, it's chemistry, it's physics. It's really interesting. So here's the lactic acid. When I was talking about crunchy sore muscles, here's your crunchy sore muscle situation.
2 terabytes and it's created.
Yeah.
SSD.
I'm so tired of At least they're not all political campaigns like they are some years. That's all.
It's one of those basic ideas you hear scientists repeating all the time.
I love Hank, right?
Correlation does not imply causation. Just because two things happen together doesn't mean one caused the other. Well, back in the 1920s, they ignored their own advice, and the result was a major misconception that's still around today. Because despite what you probably were taught in high school, lactic acid does not make your muscles hurt when you exercise. In fact, it might actually help. It all started around 1920 with a German biochemist named Otto Meyerhoff. He helped figure out many of the steps of glycolysis, which is the series of chemical reactions that cells use to make energy from glucose, or sugar. He also kind of fit the mad scientist stereotype. If you've ever seen a scientist in a movie electrifying disembodied frog legs, well, Meyerhoff did that in real life. He took frog legs, just the
Who else
leg
did it?
parts, and he gave them electric shocks.
Galvani.
Jump and twitch. But
Yeah.
after a while, they stopped jumping.
Great.
Meyerhoff found that the legs were full of lactic acid, or more accurately, lactate, which is lactic acid minus a proton. He and another scientist, Archibald Hill, correctly figured out the steps of glycolysis that led to higher levels of lactate in the muscle. But Meyerhoff also assumed that the lactate buildup contributed to muscle fatigue, stopping the frog legs from jumping around. When you exercise, your body uses up its main source of energy, ATP, within a few seconds. Then it starts to make more using a bunch of different chemical reactions. And during a hard workout, your muscles start to rely more heavily on reactions that also produce more lactate. When you exercise, the pH in your muscles goes down, meaning they become more acidic. That's called appropriately muscle acidosis. The drop in pH may be part of what causes the famous burn while you're exercising. There was a clear correlation between the lactate buildup and acidity in the muscles, and Meyerhoff assumed causation too, that lactate was responsible for lowering the pH in muscles. This became a generally accepted hypothesis, and you still see it all over the place today. Unfortunately, as we all know, assuming causation can be risky. And in this case, Meyerhoff was wrong. Even though the lactate idea was well accepted, there wasn't much empirical evidence for it. And over the years, researchers started to question whether it was accurate. Around the second half of the 20th century, they started to pick apart what was really going on. One important clue was the fact that unlike lactic acid, lactate isn't an acid. That missing proton means it's a base. It accepts rather than donates protons. Meyerhoff's idea assumed the reaction that produced lactate also produced a proton at the same time, making the muscles more acidic overall. Turns out, it doesn't. The series of reactions that generate more ATP in the muscles do produce extra protons, which lowers the pH in your muscles, but the extra protons don't come from the step that generates lactate. And in fact, lactate bonds with some of those protons, making the muscles less acidic. Lactate also isn't involved in delayed onset muscle soreness, the pain you feel a day or two after a hard workout. Scientists think that comes from microscopic tears in your muscles, although they are still investigating that. It's weird that we don't 100% know, but we don't. So despite what you hear from personal trainers everywhere, and sometimes even see in textbooks, there's no reason to be so hard on lactate. It's just trying to help. Also, a side note here, you're gonna wanna tell people about this when they say, the lactic acid, do it, but try to be in the least pedantic way possible. We have to be accurate, but don't be condescending. Thanks for asking this lovely question, and thank you especially to our community on Patreon. Your support keeps these answers coming.
So Hank has another reason, right? Hank says, not so much the lactic acid. You're going to find conflicting thoughts on that, clearly, but he has a very good explanation for why it might not be the sore, crunchy muscles, though despite some are going to say it is the sore, crunchy. The correlation causation thing is huge, right? It's huge. It's very important. That is what we're dealing with right now with the Tylenol debate. Like, is it really causation? Is it really correlation? We really need more studies to nail down exactly what's going on. There is evidence that there is some causation, but there's also evidence that it could be correlation. One of them being, why are you taking Tylenol if you're pregnant? Maybe you have a fever and that's why you took it. So is the fever the cause? We don't know. We don't know enough to be like 100%. But like with anything else, it's one of those, ask the doctor, take as little as possible. Yes.
Dr. Mike has a good video on that.
Dr. Mike? Okay, cool. There's a lot of people that have been, I've read so many interesting and conflicting study summaries and whatnot over the last, couple days. It's been pretty interesting. So, all right, let's look at these. What's your definition of photosynthesis? What you got there, Eunice?
When you convert
Stop. Let him talk. Quit.
when you convert H2O into glucose sugar.
With sunlight. So it's the process by which plants capture light energy from the sun and convert it into sugar. All right, Yusuf, what's your cellular respiration definition, since you're over there giving your brother a hard time? Oh yeah, what is it?
No bacteria.
Oh, wait, you don't, oh, he's not ready. Oh, was that good, Eunice? Was that good? Do you approve? Okay, so we're looking for the process of producing ATP energy in the cell from oxygen and glucose, releasing carbon dioxide and water, and then fermentation, we're going to say the breakdown of sugars to make ATP energy in the absence of oxygen, which is anaerobic. So what's the formula for photosynthesis? Oh, come on, you know it, because we just looked at it a bazillion times. 6 CO2 plus 6 H2O plus sunlight yields 6 C6H12O6 plus 6 O2. So then based on that, cellular respiration formula is going to be Eunice.
C6H12O6 plus 6O2 equals 6CO2 plus
You're right there.
yeah,
6.
6H2O.
Right. And instead of sunlight, we're going to see energy is not absorbed as sunlight. It is released as what? Do we know?
H2O. Oxygen.
Energy is released as heat. That's why you're warm, right? And then fermentation, you got 2 different types, so it can look different. Where does photosynthesis occur? Where?
Chloroplast cell thing.
Specifically where in the chloroplast, do you remember?
So like little round.
The thylakoid membrane. Yes. Okay. And where does cellular respiration occur? Claire?
Mitochondria.
In your mitochondria of a plant or animal cell. And where does fermentation occur? Do you know? You're unsure? Do you know?
In the lactate.
In the cytoplasm.
In the
In the cytoplasm of a plant or animal cell. So our reactants and products, our reactants in photosynthesis without their chemical formula, just what are their names? Yeah, give them to me.
reactants photosynthesis is probably
It's right here.
what you breathe.
What are the reactants? Look right here. Reactants and products. What's that?
Carbon
Carbon dioxide.
and H2O.
And what is that? Water. And then what is this? C6H12O6. Glucose and sugar. Very good. All right. How about for cellular respiration? What's it going to be? Somebody at the back table. What's going to be? This one's easy because all you have to do is look at the one before it and flip it. Flip it. Oh my gosh. This is so hard. All right, Xander, what is it? Yeah, there you go. That's it. Whoops, I clicked that one early, and that's going to be different depending on what you do. Very good. All right. How is photosynthesis related to cellular respiration?
They both work to provide energy for a cell.
That's one way. How else?
They're opposite.
They're opposites. Yeah. So they're opposite processes, but yes, they both are providing energy or creating energy. All right. And how about cellular respiration? How's it related to photosynthesis?
It's the same.
Yes,
It's
same
opposite.
opposite process. All right. And now how about fermentation? How is it related to cellular respiration? So it's going to be that all living things produce use both processes to create energy. And remember, the first several steps are the same as well. So what's your energy source for photosynthesis? McKenna! Your sun, your sunlight. What's your energy source for cellular respiration? Yeah, food, sugar, basically is what we're doing. Yes, sugar. All right, and then what is your energy source for fermentation again? Yeah. Well, you're producing the ATP. So it's, again, it's food. Consumers. So yeah. Okay. All right. Lastly, we're wrapping up a little early, which is fine because some of y'all can help. Oh, who can answer that? Caitlin can answer it. Why is October important?
It's Tinker Day.
What is Tinker Day, Caitlin?
It's the best day ever.
Explain for our newbies why Tinker Day is the best day ever.
It's because you get the donuts.
We get donuts? What is Tinker Day?
It's from Holland's University. It's like
A day we don't
Tradition.
invest in anything.
and then you climb up to the mountain and sing a song.
What is people are like, yes, you're getting it right.
And you get fried chicken.
Get fried chicken. Tinker Day is a tradition. Yeah.
And you get to wear funny costumes.
Yes. Tinker Day is a tradition / holiday at my college where one day in November, and nobody knows when that day will be except the president. There's one day in November and she will declare it is Tinker Day, and she doesn't declare that until 8 a.m. that morning, or actually it's more like 6:30 a.m. that morning. And when it's Tinker Day, all classes are canceled. It doesn't even matter if you had a test, whatever, all classes are canceled, but you have been preparing for this since you arrived on campus in August, so you are ready. You wear your craziest outfit, but before you do that, you go down to the dining hall in your pajamas and you eat all the Krispy Kreme donuts your tummy can handle, along with whatever else is for breakfast, but it's always Krispy Kreme donuts. Then you go back to your dorm and you don your very carefully constructed, curated costume. And then you walk out to the front quad with all of your sisters, and you're very excited because it is Tinker Day and the president reads a proclamation, and then you walk up a mountain dressed like a crazy person, where at the top you sing songs and you play games and you do skits and you eat fried chicken and you eat Tinker cake, and then you come back down and your evening classes resume like after 5 p.m. If you have rehearsal for choir or something, you still got to go, but the rest of it is done. Now, Tinker Day means something for you. So it will be in your email. Be sure to read it. It is the easiest way to do better on your midterm. Okay. These points go to your midterm. So what you will do is you will either dress up or go for a hike or eat Krispy Kreme donuts or do all three, because I will tell you when it's Tinker Day. Or if Tinker Day happens to be on a Tuesday, you get to do what? You get to come to class all dressed up. And it has happened before. It has occurred on a Tuesday before. Regardless of when Tinker Day falls, I will give you Krispy Kreme either that day or the next day. The next Tuesday, whenever that is. So if it's on a Wednesday, when you come back next Tuesday, there will be Krispy Kremes waiting for you. You do not get out of class, but you do get donuts. So for some reason you cannot have Krispy Kremes, let me know. And I prepare. I have learned over the years, you can buy your donuts early and pop them in the freezer, and then you just put them in the warming oven, and they're perfect because they're nice and warm. So I already have a freezer loaded full of Krispy Kremes right now because I took care of this. So other announcement regarding Tinker Day is I host one of 2 Atlanta area Tinker Day parties for the college. And mine is Friday night. So ladies, sorry, gentlemen, it's a women's college. Ladies, if you want to come Friday night with your parents, you are welcome to come to the Tinker Day party. We have, I think there will be 4 of us of RSVP alumni so far that are here. So it's usually a small group, but they're a fun group. I have 2 ladies that come every year. One is the class of 1976, which is the year I was born. She's a lawyer. She's a lot of fun. The other is class of 1986, and she is an absolute riot. She's a realtor and an artist, and like the lawyer lady brings her husband, who talks to my husband. The realtor slash artist is like gleefully divorced, and she's a ton of fun. So they come every year. I got another girl coming from 80 something. I don't know what. I did have a class of 23 up until last night, but I think she dropped. But gentlemen, if you have a sister, you can send your sister and your mom or whatever. But we have fun. We always enjoy it. And it's good times, and I will have fried chicken and I will have Tinker cake, because if you host a Tinker Day party, they send you the secret recipe. So secret recipe for Tinker cake, which I have tried to bribe my classmates with in the past. I'm like, oh, well, I know a couple of you live in Metro Atlanta, and if you want Tinker Day cake, you're gonna have to come get Tinker cake, and then I'll give you the recipe card. So it's fun. So feel free to come if you want. And since we have a few minutes, if you drove, you can leave. If not, I'm gonna try and find that Appalachian lady that makes the sody pop. You can watch her. She's fun. But y'all are good to go. Turn this off though.